Today's Tech Short, we're talking to Dr. Nathan Brown, a computer scientist turned chemist who's spent his whole career working on how we can design drugs better, faster, and more intelligently. He's an award-winning scientist and author. He's a fellow of the Royal Chemistry Society, and an inventor whose work spans chemistry, computing, and creativity. He's a highly creative person. He's got a lot of views on how AI and computing intersects with chemistry and with drug design. And he's led research groups in academia and worked in startups, at the center of the AI drug discovery storm, as it were. So he's a really interesting person to talk to. I hope you get a lot out of the conversation. Nowadays, he's working on AI tooling, and we'll get into a bit of that towards the end of the conversation. I hope you enjoy it. Nathan Brown, welcome to Tech Shorts.
Thank you.
So you and I met a few weeks ago in London at an event that the Alsace flight team put on, which big shout out to Christian and Will and Steve and all the guys who put that event on. It was a really good day. But your talk around drug discovery, drug design, and its computational roots nowadays was really interesting, which is why I really wanted to have you here today to talk about it. But the most remarkable thing I found out in talking to you afterwards was that you're actually a computer scientist, not a chemist or a biologist. How did you get into this? How did you wind up where you are?
Yeah, that's a good question. I'm evolutionary algorithms will be the obvious answer for me. I got interested in genetic algorithms in my later high school years and then undergrad, building predator-prey models that were evolving over time. And then one of my friends, he's now a scientist up at Manchester, professor there. He was in biochemistry at the time, and he was one of my friends in Aberdeen where I studied. And he said, "Oh, this guy's doing these genetic logarithms you like." And I went, "Okay, I think you mean algorithms." And he was looking for a PhD. He's looking for a PhD student. So I thought, okay, I know nothing about chemistry. So I contacted Professor Peter Willett up at Sheffield, saying, would I be of interest? My background, he says, yes, definitely. I've got a really interdisciplinary team. We've got chemists, but we've also got mathematicians, statisticians, computer scientists. It's in the school of, at the time, information studies, because that's historically where it came from, by chemistry librarians. So that's how I got into it. And I ended up writing graph matching algorithms using genetic algorithms there. For me, chemical structure was just another data type. It's just a acyclic weighted graph. That's the representation. So it's very easy for me, coming from graph theory, to deal with those data types.
My God, the computer scientists are just going to reduce the world into directed acyclic graphs, aren't we?
Exactly.
So we're eventually going to get to those genetic logarithms, I think. We'll get back to that in the discussion. And it may well be very soon. So when I my training was in physics. And I aborted my PhD so that I could go and travel the world and do exciting startup things and whatever. But when I was working in physics, to me, the biological sciences and chemistry and so forth, I couldn't, the most compute that those folks did was sending emails or writing a paper in Word on these very nascent things called laptops at the time. Right? So I'm kind of showing my age here, but fast forward 20 some years, and here we are in this world where, genetics, bioinformatics is a field. The amount of compute being thrown at this is enormous. Where does that fit in drug discovery nowadays? Because drug discovery is it's a big, fast moving scene, right? And a lot of stuff is changing. You're at the heart of all of this. You're sort of like in the center of this storm. Where does all this compute fit in that stuff? And what are the big bits that have changed? But I'm kind of interested in understanding the bits that are not changing, the stuff that's also hard that we still haven't cracked yet.
Yeah. So the main aspects that have really changed over the time I've been in this field, which is over 25 years now, are access to data and access to APIs, algorithms that have been implemented open source. So biology was a bit ahead of the game in the 90s using, back then it was Perl, a lot of string-based, so sequence-based analysis using Perl. Obviously, our primary data structure in chemistry is a molecular graph structure, so it requires a bit different kinds of algorithms. I'd say two things that have significantly changed from when I did my PhD, where I wrote everything myself in pure ANSI, all the memory allocation was my own. That software ran in tiny memory footprints, like 2 megabyte memory footprint, doing molecular design.
It's the only real way to do it. I'm right there with you, baby.
I say that as well, but sometimes I start thinking I'm just this old guy who's you should go through the misery I went through, because that's the only way you learn. But
that's not true.
No. But two things I'd highlight, and I use them I was using them before this call. Kemble from the MBEL EBI in Hingston, outside Cambridge, they released probably 15 years ago, because I gave a talk at the symposium for the 50th anniversary last autumn. It's a huge data set of basically all the data has been mined from the literature. They've got patents as well, data, and just getting that open source out to the community to use. Before 2000, what would that be, 2010, 2009, we had to beg for data from Big Pharma, and they had very small data sets that they were willing to share. And nobody had abstracted all this data out into machine-readable form. We did a good job as scientists to hide the data in PDFs that we had to then reverse back out. Yeah. Right. And it's all image data. We didn't do, if you did supporting information, so it was, again, PDFs, image data, and the numeric values were all very difficult, quite often scanned rather than even typeset. Yeah. So it's very difficult to extract. And I remember having to hand rewrite all these chemical structures and getting them revalidated, because you make mistakes when you're manually copying stuff. And the other one would be the RD kit. So this is a good friend of mine, Greg Landry, open sourced when he joined Novartis around about the same time, the late 2000s. And the RD kit's it's a C++ API, but it's got Python bindings. So you can do a lot of really powerful chemistry work very quickly. It's in Python at the main interface that most people use nowadays. So it has its challenges in terms of efficiency. And when you're working with billions of data points, it's quite difficult. Sure. But they're the two things.
But the ability to just develop code quickly in Python is amazing, right?
Yeah. It's by far one of my favorite languages, just for, if you know Paul Graham's work and his book on hackers and painters, I love just doing the scientific coding where I just wonder what's happened. What happens if I do this? What happens if I try that?
I sort of wonder, a lot of those things were designed for, like Python is a great fast prototyping language, and if you're getting moving fast, and if you've got an idea and you want to get moving fast, it's perfect for that. Right? Yeah. But of course, maybe fast forward in 20 years to the future from now, or maybe even 5 years, the AIs will be doing prototyping for us, and they'll be using languages that we don't need a language for human convenience. We'll be needing a language for, bot convenience, probably. Yeah. That's almost certainly where we're going to go. So, one of the things that I admired about the life sciences when it came to computing is that because they came to computing late, they adopted really good modern software engineering techniques. I'm not going to call Perl a good modern software engineering technique, but things like containers and Git repositories, and so version control code. And and you look at the in the bioinformatics world, which I have a lot of activity going on in. They use Bioconda, and so they they have thousands of algorithms in packages in Bioconda, so that they're all shareable. And I mean, they've taken a modern approach to sharing, and there is a ridiculous amount of sharing and collaboration going on. It's kind of good. But you're saying open data was also a pretty significant aspect too. What are the big data sets that are important here? And more to the point, how big are these?
Yeah, still not big enough. And we a lot of my friends complain that Big Pharma's got all this data, but they might have it, but they don't know what they've got, because it's all over the place within these large companies. And these data, basically, most commonly it's the chemical structure, and we use something called a SMILES notation, which is a linear or line notation for the chemical structure. Very simple encoding written by Dave Weininger back in the 80s. And associated properties. So they might be a biological activity against an isolated enzyme of interest. They might be solubility or Do they pass through the central nervous system to get into the brain, for example? All these kinds of properties are out there. And then we can build molecular descriptors or featurizers of those chemical structures, and then start building models of basically the physiological action of this molecule as a function of its chemical constitution. So it's a function of how it looks, how it's made. And it's interesting because that was actually coined by Alexander Crum-Brown in the 19th century as what I call the first predictive modeling equation in chemistry. But it's taken us many more years to actually do that in practice. And this field of this predictive modeling for chemistry called quantitative structure-activity relationships actually came out around about the same time as artificial intelligence was coined in the late 50s, early 60s. So we have been, without computers, we have been thinking about a lot of this stuff for decades. But it's only, as you say, relatively recently, that we've had the compute power to do a lot of the thought experiments in actuality on these machines.
And some of these experiments are becoming huge. I mean, I've worked with customers. That do a lot of molecular dynamics, and these things like NAMD and GROMACS and all those other codes that for simulating what's going on. But they measure progress in nanoseconds per day of progress, nanoseconds of simulated time per day of clock time, and that's it can be grindingly slow to just using a massive amount of compute just to simulate one small molecule floating in water, et cetera, et cetera. That's changed a lot with AI, right?
Yeah. I do more of it the other way, where so that kind of simulation work is you're trying to find a lot of information about small systems, relatively simple systems, but still very complex systems. I do a lot more work around the exploration of the chemistry space. What's the available small molecule drug-like chemistry space that we could realistically sample and evaluate in silico? And if you think about combinatorial explosion, and I've had chemists swear at me before when I explain to them how big the chemistry space they're working in, you're normally making a few hundred to a few thousand compounds for any particular drug discovery project. This is in a potential data space of there's various estimates of like 10 to the 20, 10 to the 100. It's so big, it doesn't actually matter how big it is. It's just unrealistically big.
It's effectively infinite,
Yeah. So we're trying to evaluate in that space. So we're using, we can come back to genetic algorithms. That's where most of my work has been on for molecular design. So you're sampling and small enumerations out of that space, and then scoring them, which is by far the most computationally intensive part compared to the graph construction. And then feeding those back in as the GAs work. And now more recently, we've got more into large language models for user based on the smile strings to build new compounds, new molecular structures, and making those predictions themselves. And I've been doing a lot of work on that quite recently, using LLMs for a lot of the work I've done differently in the past. I'm now using LLMs to achieve that.
And so when you're talking about using LLMs and GAs to go there, how many are you dialing up the number of compounds that you're exploring by what order of magnitude here or more?
It's more the region of the expanse of the chemistry space you're able to cover by doing this sampling rather than enumeration. So there's some companies I know and academics are doing large virtual screening. So virtual screening is in silico prediction of what a chemical structure is going to achieve potentially in a living system. They're looking at data spaces of 10, 20, 30 billion chemical structures that are realistically synthesizable based on their knowledge of synthesis, and they can turn around. So I'll give a shout out to Enamine in Ukraine, who do a lot of this kind of work and pioneered it. They've got very robust synthesis plans. But that space, you can build that big space. It's unenumerated, so you can again sample it by looking at this core scaffold of the molecule, and then what we call R1s, these substituents, and R1s, R2s coming off. But it's still so big, we didn't have the systems to search that. So we've had to do the computer science work, and they work with clients in Hamburg on called Biosol IT that built some software based on an algorithm developed like 25 years ago by Matthias Rari called feature trees. But even though it's still 10, 20, 30 billion chemical structures in there, it's very quickly you find everything that's of interest to you in maybe 100 or 200 molecules you can get made and tested. Right. So it's still only scratching the surface of what we can deal with. Whereas,
but presumably that's a better selection of 100 or 200 molecules to start with rather than what you might describe it otherwise.
Yeah, it is. I mean, mainly because you can actually realize those molecules. They can get made and then tested in laboratories, which is not always the case. Believe me, with some of the computational algorithms I've built, they come up with some very wacky ideas of chemical structures. And that's where I just hold up my hands to my colleagues who are chemists and say, I'm a computer scientist, I don't know what I'm doing.
Don't blame me.
Yeah, let's figure this out together. Although we have improved with some computational methods for synthesis planning. But it's very, I think, so it's very quickly to exploit that space, and you have to be aware as well that everyone's mining that space because everyone's dealing with the same problem. So very quickly you get very encumbered space that many other people are working on the same region of that chemistry space that you're working on. And then that goes down to intellectual property rights, who can patent first the chemistry space you're working in, so that they can protect their invention and others can't use it, or they can use it, but they can't necessarily commercialize it.
That's really, it's an important aspect, because if you're going to go and do all of that work, if you're going to do all of that work and so forth, you do need to be sure that you're going to be able to profit from it at the end and commercialize it successfully. Okay, that makes sense. How big like to zoom out, how much impact are all these techniques having on this space? You know, am I going to walk down to the pharmacy today and, or to the drugstore and buy pills that have been produced from this process already, or are we still waiting for some of these to come through?
Yeah, I think any modern drug approval has been touched by computational methods, whether it's on the biology or the chemistry. Maybe not fully, well, definitely not fully automated, and maybe not as augmented with the in silico work as we'd like it to be. But it takes time to change how we work together. And an additional challenge of this is people coming from different disciplines where they don't speak the same scientific languages, and they're having to find translators. And I typically am one of the translators because I work at the boundaries between computer science, AI, ML, and chemistry and biology and clinical work. So we quite often in teams I've led in the past in industry and academia, we tend to be the ones who can help people communicate with each other. So I think everything is being touched by it now. Chemists are now using these methods routinely on their desktops. I'm a big fan of democratization of tooling with the appropriate guide rails to make sure that people can refine their hypothesis without me being the gatekeeper. Sometimes I would need to be the gatekeeper because I've studied this for my career, so I know where the bodies are buried, for example, what's going to come up to bite you. But giving out to the chemists and whoever else is working on the project team, and I've got some wacky concepts around citizen scientists being involved as well. So there's no reason it has to be an expert, because it's just you're solving a problem.
The astronomers have been doing that for some years, citizen science, right? And using the web for the way to actually interact with all of them and get them to work and deploy packages of tasks to them, and it's worked out really, really well.
I'm actually working on an app at the moment to do that as a alpha kind of go, a quick go rather than alpha go, a quick attempt at trying to do the citizen scientist work for chemistry and drug discovery. Initially, I'll be crowdsourcing from expert chemists, but ultimately it'll be something around not really around about gamification. So I have worked with a company in the past as an advisor on gamification of drug discovery in Barcelona, and they had some really cool concepts. So I quite like to further those kinds of.
It'll give chemists something to do while they're sitting on the loo rather than TikTok.
Yeah.
There you go.
Exactly. Yeah. Or waiting for their computer to boot up in the morning. That's often what my colleagues do.
I was having a discussion the other day with one of our product folks who's been working with. He's been working with they're called the Toyota Research Institute. They're working on Japanese company working on robotics, right? And they're working on essentially an LLM for the robotic space, so that they can do anyway. It was an interesting discussion, but what was most fascinating about it was we'd achieved something kind of big for them because through just tooling changes. We'd connected Batch with SageMaker, which is SageMaker is the environment you use on AWS for building your model environment. It's all Jupyter Notebook based, but sometimes you just need to run a big training run, or you need to explore 25 different possibilities quickly. Mm-hmm. So you need something like batch to actually just deploy a lot of workload to the GPUs quickly. So anyway, the details are unimportant, but we've done this integration and it really enabled these guys to get moving faster. But the key innovation was that they're still able to do everything inside Jupyter notebooks. Mm-hmm. You didn't have the robotics folks who were really comfortable working in a Jupyter notebook, then didn't have to leave the Jupyter notebook to do something bigger and faster and better. You just gave them a few extra pragmas in Python and they could just get busy. Yeah. So how important is that? How important is this sort of general leveling up of access to the scientists? How much of an impact has that had? Cloud is sort of part of it, but HPC generally as a technique is part of it. How big is this? How much of an impact has this had?
It's had a huge impact because it increases the adoption of these approaches rather than, I'll go and speak to my friendly computational chemist and they'll do some docking where you take a small molecule and try to dock it into protein binding sites on the computer. They can do it themselves. And then we learn a lot as humans by making mistakes. And it's okay to make mistakes, especially maybe not in the lab because that's a bit more dangerous, but in the computer, you can try things and experiment with ideas. So it increases the adoption of the methods and it takes away the fear of the unknown of these approaches, because the computer will always give you an answer. That's how they're set up, and that's the problem people are facing now with things like LLMs and ChatGPT. You always get an answer, but it's up to us as humans to decide: is this a good answer? Have I asked a good question? What? And then, now I realize I see the answer. I realize my question was wrong. So let me refine the question, and you can do those iterations and then learn. You have the human in the loop, literally. Doing that as a human expert in the loop. And a lot of the work I've been doing in the startup and the private sector the past 10 years or so is developing these platforms that enable people to do this seamlessly and then drill down on the molecular design level, drill down into models for everything they're interested in for this particular project, but finding, has this molecule ever been put into a patent, or is it available in the literature? Is it available to purchase from a vendor? You can start joining up all these data sets together. Has it been expressed has it been demonstrated to bind in a protein structure? Is there an alpha-fold structure for that protein? All these kinds of things, or PDB structure. All these kinds of things, you're joining up the data. You can start with your entry point of your chemical structure or your protein, and you can start navigating, hopping between all the different data types, from chemical structure to protein structure to synthesis data to genetics. You can start navigating between these spaces. And we've done a pretty poor job of it historically because we've all worked in our relatively siloed fields with the interfaces. Now we're blurring those interfaces, and we're overlapping or dovetailing between the disciplines. So we're improving that cross-connectivity and cross-pollination of ideas. But it'll still take time because it's changing the way we're thinking. But I did a quick look last week. So a friend of mine in Nottingham set up a digital chemistry master's program. And digital chemistry is what we now call this kind of area, anything computational with chemistry. Yeah. And I thought, it feels like there was someone else who set up one of these master's degrees. And then I did a quick search, and I found there's 9 or 10 set up in the UK starting next year. So I'm thinking, because part of my role with the Royal Society of Chemistry is helping understand what the community needs as well. And thinking, is there a room here to bring all these cohorts together from different universities to understand there's a huge community, an ecosystem here that they can learn from and build on that. Mm. And then looking at that, you're now getting chemists who are, and I'm involved with one course doing Python for chemists, so learning to code. And coding is sequence, selection, iteration. You can learn it in 5 minutes, but really learning how to solve problems you face is a really good way of learning the coding, as well as learning the environments, the APIs that we're dealing with. So it's a really great time for the field that's only really changed in the past few years.
It's interesting. I mean, your point about, we learn by making mistakes. It's an incredibly important part. I often remind everybody, because we've got a very different approach to doing HPC in the cloud, right? It's elastic. Mm-hmm. It's a synthetic thing you create in the cloud. It's not a hard physical form that you have to go and take years to build by doing tenders and RFPs and contracts and pouring concrete. I mean, we did all of that already. Yeah. But I keep reminding everybody, with this sort of synthetic environment, you're only 15 minutes away from getting back to where you started if you screw something up.
Yeah.
And as engineers, we learn by breaking things. Like I swear every kid who goes on to become an engineer was once a child who took a screwdriver and pulled something apart and couldn't put it back together again, right? And that's how we learn. So I guess that democratization aspect is really that we're giving people more access to break more things. It's a perverse way of putting it.
No, absolutely. I was that kid who took apart the record player in the 80s when my parents were out in the kitchen or in the garden, and then they come in and they see it all, this massive thing disassembled.
Were you able to reassemble it, or was that a learning?
Yeah, luckily my dad was an engineer, so he managed to do that. But he was called himself a mechanic. He didn't like to be called an engineer.
And he probably quietly appreciated what you were trying to do, even though it may have been seething with anger.
Yeah. But it was my father who got me into computers at an early age with the ZX81 and ZX Spectrum and in the early 80s when I was very young and shouldn't really been coding. I call it coding, but I was like changing the color of the border and that kind of things on the old Spectrums.
Those early machines were really important. I had a Commodore 64, and I think I mentioned to you before that, those learning assembler on a 6510 processor. Really good instructive stuff. Those were good ways. So fast forward a little bit now, and so here we are in this world of, there's genetic algorithms, there's large language models. Now we could probably get, we could probably get doom scrolling and apocalyptic about what might happen when machine God takes over, but on the presumption that machine God is still a while away, and more to the point, it's not going to solve all problems. What about the here and now when it comes to AI in drug discovery? It's certainly protein folding. I mean, the open fold, alpha fold and open fold and all of those, all of the folds. Hugely impactful, obviously. Mm-hmm. But how impactful is that? And how much of the drug discovery space is getting impacted by it? Is it just a little tiny corner? Is it just the beginning bit? Is it fill this in for me, because you know this well.
It is hugely impactful and influential, and I think one thing we're learning is what we don't know at the moment, because we've now got access to all of the human structural proteome predicted through AlphaFold and OpenFold and whatever else. But now we're trying to deal with, is the conformation, is the shape of the protein that's being predicted, is that relevant for my small molecule discovery? Or do I need to then go back to what we call a classical method like molecular dynamic simulation to refine that binding site to get it into what would be an environment to be relevant to a small molecule binding? So it's hugely influential. And I think we're starting to see it more as well in chemistry, where we've got obviously the availability of the chemical structure space, but we're starting to make more data that's augmented data sets or synthetic data sets based on what already exists, which is how AlphaFold works. It uses the protein data bank's crystal structure database for proteins to then predict the rest of the structural proteum.
And that database was put together sort of, from experimental data, right?
Yeah. And it's, I can't remember how many thousands it is now, but it's a big data set, a relatively big data set for this field anyway. And it's the same now in chemistry. There's companies out there who are mining data from big pharma companies like Genentech, Roche, and AstraZeneca. There's a company up in Macclesfield called Medchemica who do this. And then they're finding what's called matched molecular pairing in chemistry, where you've got two molecules, everything in the molecules stay the same except from one exit vector, one substituent or substructure changes. And then you have measured data for both of those compounds. So you can be able to calculate a delta. So how is that property changed or affected by that change in the structure? And then you can start simulating data sets based on those changes. So then you can start interpolating a larger space that is based on experimental data, but unknown data. And then you have confidence and uncertainty levels associated with that. And you can do this on very close to the empirical data sets with the real experimental data. And then you can start using those to build models that then you can extrapolate out and find more interesting chemistries that otherwise you wouldn't find. So all of that is helping. We're improving. There's a technique in chemistry drug discovery called scaffold hopping. I've written a book on it, where you're changing the core substructure of a molecule So you're finding essentially, I think it was finding a wormhole in chemistry space where you're over here somewhere in this high dimensional space, and you manage to get over here and find something that is equivalent in function, but structurally, chemically different. And this is very powerful for, obviously for intellectual property reasons, because scaffold hopping is a way to get away from an encumbered space in terms of IP. But it's also very powerful to mitigate some other characteristics of the molecule that aren't beneficial for generating a small molecule that's already bioavailable.
Could this be something like, sort of off-target?
Yeah.
What do they call it? Off-target binding?
There's off-targets and anti-targets, and there's things like bispecifics. So sometimes you might want to hit a few proteins. So you might not want to be exquisitely selective. So that means you only target one protein. But most drugs we develop are hitting a few other things to different levels. It's whether that ratio of potency to the other off-targets or anti-targets are beneficial or not for the patient. But there's also things around, is it soluble? So will it make a good orally bioavailable drug that you can take as a pill and then goes into the system and doesn't metabolize too quickly? It gets distributed around the body to where it needs to be. It gets excreted appropriately. It doesn't have other toxic effects. You can start building all of these parameters and models into your workflows. And I think something I've been working on a lot in the past few years is bringing those predictions further up the pipe. So we start considering them much earlier, because I use the analogy of chess. When you're playing a game of chess, it's the same in drug discovery. Your opening moves determine your end game. So you need to be really sure and explorative in your early drug discovery, your hit finding, we call it, and hit expansion region of your small molecule drug discovery space, and other new modalities as well, because you're going to be locked into that. And there's potentially a suboptimal solution, going to computer science terminology, for where you want to be. So you need to really kick the tires and everything as much as you can early on, and maybe not get too beholden to it, because you're still going to make mistakes, because it's the nature of the unknown. We're going to make wrong moves, but mitigate these potential challenges.
So this technique of scaffold hopping and just the fact that you can do so much of this computationally, right? This gives you the ability to sort of maybe look forward, to kind of finding the things that you really don't want to find out in a stage 3 clinical trial.
Yeah.
That I don't know, this drug has a horrible effect on people of Asian descent, or on people with a Y chromosome, or, you can find all that stuff out much sooner.
Yeah.
And potentially scaffold hop over to something that's got similar chemical properties, or at least biological properties, without it having quite the impact on the, on the host.
Yeah, absolutely.
So this is huge. And what role does AI play in all of this? I mean, protein folding, obviously, we know about, alpha fold, open fold, and stuff. Where else is this coming up?
Yeah, I mean, it's been used and has been used very widely for decades, going back to QSARs in the 50s and 60s. But my training was all on building models where I'd write my own descriptors or featurizers for the molecular structures. I've got the data, the potency, the IC50 or whatever, and then using those features and the Y variable, building models using random forests, partial least squares, logistic regression, whatever helps you. And you can build those models to make predictions for the unknown, with the caveats of, this model might not cover the data space you're interested in. It might have some uncertainty about the prediction. It's outside the applicability domain, these kinds of things. But in a way, that always basically tells you, like, if I don't know much about here, but it looks interesting, then I need to know more here. And that's where we need more chemists making more compounds, more biologists testing those compounds, so we can pull those back into our models to improve the data space and the models. And then it's a fortuitous cycle of doing this. We're using approaches in active learning to improve the data sets in a directed way.
I think I've got some bad news for you, because I don't think we're making enough chemists. And that's problematic, right? So I think in most Western countries, we got this demographic flip going on, this sort of inflection point where we're ending up with more retirees than we have workers in the population. Well, most of us are going to get there. Most of our countries are all going to arrive at this place, just depending on what time. But we're not producing enough chemists, right? We're just going to have a decline in the number of chemists. We've got a historically high education rate in our populations, particularly in the Western world. But that's still not going to counter the overall trend line, which is we just don't have enough young people.
Yep. Yeah, and it is telling when you look at the industry now. We are improving the processes, so there's a lot more lab automation going on where you need fewer people to do more. In the same way we have that in computer science and with AI, but you still need those people to know what they're doing, and that we're probably going to face this with LLMs being adopted so widely that people, do they need to learn to program like we did in the early days of our careers, or can that be offloaded to something in the cloud to do it for us? But we still need that knowledge about when I get ChatGPT to write some code for me, I can still go in and edit it and debug it because I trained it in that way. I get worried where the answer will just come back and then it's not actively investigated to see if it's a good or a bad answer for the.
So maybe that actually gets us back to an interesting sort of connection point back to the beginning of this conversation, right? Even though it's going to be GPTs and the like that are going to be creating code for us, Claudes and Chats and all of the rest, they're going to be creating the code for us. We probably do need them to be creating the code in human readable, a human friendly programming language, simply so the humans can inspect, debug it, validate it, to make sure that what it's doing is correct. I mean, I'm an active part of that when I do use an LLM to write code for me. I always want to see what the code is and I want to finagle with it. But maybe so regardless of whether machine god comes or not, that's still going to be a hugely important part of it. I think that I would definitely argue that the need to be able to punch out hundreds or thousands of lines of code myself is no longer there. I don't need to do it. I can get a machine to do that for me. As long as I can validate it, it's okay. But it's a matter of figuring out how to increase the human productivity. It just goes back to that, your point. If we are going to have fewer chemists, we need them to be more productive and more powerful.
So there's a lot of mainly academics working in this space about lab automation and having robots to generate the compounds. There was a company called Cyclofluidics that did it about 15, 20 years ago. There's companies in the US, Stratios, I think they're still around in La Jolla. There's Lee Cronin up in Glasgow. There's Imperial doing this with their lab automation in London. So there's a lot of this kicking off now. But it still feels some way to go before this is truly automated, because it's still quite often it needs the humans not to be thinking, it needs the humans to be cleaning the machines so they don't gunk up with all these reagents, etc.
Ouch.
That's what I hear from my colleagues in these areas. So that's what we're good for. We're the maintenance crew to go in and
It's not quite as bad as that. Well, a friend of mine pointed out to me a while ago. He said, we sort of had this dream of computers taking away all of the crappy jobs from us so that we could have more time to spend, creating music and writing poetry. But now the machines are writing the music and creating the poetry and leaving us with the crappy jobs to do. So we need to get that sequencing right.
Yes, for sure.
So what do you think is the future here? And I don't know whether we want to wax too much around, when machine god and an AGI comes along, but what do you think is the immediate future and the real trend lines here? Where are we going with all of this? How fast, how soon is this going to have an impact? Because creating drugs is not just a computational process or a chemical process. It's a huge industry and a massively well regulated, or is it well regulated? Anyway, it's regulated.
Yeah, it's definitely regulated.
It's a massively regulated process. So there's a lot of flywheels here that are not gonna change speed quickly. Yeah. What are we likely to see in the next few years, realistically?
Yeah, well, I mean, I think we're already seeing it throughout the industry is the take up of LLMs. Everyone's using those for various reasons. I like you just said, I'm also using it for coding, and the RD kit is part of ChatGPT now. So you can get it to write the code and test the code, and then still give me the code so I can do my stuff with it. Right. So I'm using it for coding, but using it for writing first drafts of reports or emails, etc. This is streamlining as we've all got our AI assistant now to do this kind of work, or an intern, depending on how much confidence you put into the output you get from some of these tools. So I think that's clearly happened, that maybe changed much faster than people anticipated. It's only been a couple of years, really, that's seen wide adoption. I think more fundamental things that are going to improve is access to data, the generation of the data with open software. So you talked about like things like OpenFold, OpenBind. There's OpenAdmit now. One of my friends, Pat Walters, is heading up to get these data out into the community that might be in pharma or might be generated as part of their funding, but use those data to then build models that are also shared to the community in a pre-competitive sense, so that everyone can improve their modeling approaches, and you get a user base who are testing everyone else's models and finding limitations and then sharing those back to the community. So this is starting to happen now, where the data is being generated more widely, more openly, rather than being locked in historically hard disks in big pharma offices. I had to harvest data at Novartis when I was there in the early 2000s in Basel. I had to cycle across the Rhine many times from the two sites to get data off people's hard disks that they had in Excel spreadsheets. They were the databases, the enterprise data lake.
Hard drive sitting in the third drawer of the filing cabinet at the back, and that's the stuff.
Exactly. Yeah.
We've all been.
And no backup strategy, all that kind of. No backup at all, let alone strategy.
Yeah.
So this is changing, and as I said earlier, it's joining up the dots of those data sets. How can we navigate between different modalities, from small molecule drugs, protacs, macrocycles, up to antibodies and other things like nucleotides, etc. But also between the chemistry and the biology, going as far as the clinic, mapping into genetic work as well, bring this all into one-stop shop of data that you can navigate as you can imagine like a Minority Report kind of environment, where the data is all at your fingertips. You just have to ask your question and see what the data is available, but also probably having some kind of agentic AI also doing some navigation for you by learning what you're doing and then bringing things back that may be relevant that you hadn't thought of.
That kind of brings us full loop, because we opened this discussion where the thing that was really making the big changes in computational chemistry and drug discovery was the accessibility of data and the accessibility of tooling and the democratization of that. But it sounds like the real constraint now is still the accessibility of data and the availability of a large amount of data.
Yeah.
Interesting. And is that data? I mean, I would gather that an awful lot of that data is still going to be very proprietary. Novartis is going to have a bunch of that data, and they're not going to want to share that, or at least not all of it, because it's not in their interest to do so.
Yeah, absolutely.
What's sort of the dividing line here? Like, you've worked in these large pharmas.
Yeah.
How much of the data have they got? I mean, you made a comment earlier. I think people accuse them of having a lot of data that they don't know that they've got. Is that true?
Yeah.
Probably is.
It to an extent, it's true. It's much improved over the past 20 years since I was in some of the big pharmas I talked about earlier. So the data is largely available through centralized databases and searchable by everyone in the company. So the data is there, but the data will be biased towards what therapeutic areas those companies have worked on. So if they're more neuroscience focused, then they'll have different kinds of chemistry spaces they've explored because they want to get past blood-brain barrier. All sorts of different therapeutic areas will indicate the kind of chemistries they've explored.
Can that give them a blind spot, like in the sense that so they've only got data around, say, I don't know, endocrinological functions, hormone functions and stuff, and they don't realize that when they go and stick a particular drug into a clinical trial, that it's going to pass through the blood-brain barrier and start having neurological impacts. I mean, so is it worth their while? Is it sort of necessarily in their interest to start sort of sharing some of their data so that others might share their data so they can explore this stuff?
I think that in an ideal world, that would be where we are. I feel when speaking with people in the industry, there's a reluctance because they don't necessarily know the value of the data they've not found useful that they've generated in-house, but they don't want to give it away in case someone else discovers the value of that data, and then it's nobody's anymore because it's out in the public domain. So there is a reticence there about giving away what could be the crown jewels. And if you look historically, a lot of drug discovery has been getting lucky, but knowing when you're lucky to capitalize on that. So, I mean, Viagra is a good case in point. It wasn't developed for erectile dysfunction. It was developed for angina and other conditions, but it had this side effect.
Remarkable property.
In the trials, which is hugely enabling and empowering for men who have erectile dysfunction. Well, one of my colleagues in my former company, Dave Brown, he was one of the inventors of Viagra, and it's fascinating stories he tells about how important that was to people, but it wasn't what they were aiming for. And there's lots of examples of that. So I think
target finding.
Precisely, but it's interesting because it just shows that as scientists, people often think we have all the answers, but the whole point of being a scientist is understanding the questions you want to ask. We don't have the ansers.
I think it was Feynman that coined this, that the happiest state for a scientist is confused. He always found his days much more enjoyable when he was in his lab with a whiteboard, with a blackboard, as it was, when he didn't know the answer and he was confused by something. That was a delightful day for him. So well, let's hope we can have many more confusing days and this productive stuff that comes out of it. Nathan, thanks for coming along and talking to us.
Oh, you're welcome. It's been great fun.
And you've given me some food for thought, because I'll be having a conversation in a week or so with a guy who set up our open data program in AWS, and he's since gone on to greater and more wonderful things, but he's still got a really big thing about data. And but he wants to see, he likes to think of data as, instead of really thinking about it as open data or public data, he wants to think about it as data products. Yeah. He said when you put that word after it, you put that product word in there, it forces you to think about how it might interact with other firms or other people or other users. It forces you to think about its value. So I'm going to take some of the ideas you've given me. And
fantastic.
So thanks for coming along today. Appreciate the time.